The Autism Research Institute relies on the generosity of donors like you to make this webcast possible. If you enjoy this presentation, please consider making a donation. Thank you.
Now, before we get started, I'd like to introduce our speaker. Dr. Pamela Lein is a professor at the University of California, Davis School of Veterinary Medicine in the Department of Molecular Biosciences. Dr. Lein received her Ph.D. from the State University of New York at Buffalo and her MSEH from East Tennessee University. She has previously held faculty appointments at Oregon Health Sciences University and at the Johns Hopkins Bloomberg School of Public Health. Her areas of interest include cell and molecular mechanisms of developmental neurotoxicology, gene-environment interactions that influence susceptibility to neurodevelopmental disorders, and the role of autonomic nervous system in environmentally-induced asthma and cardiovascular disease. These webinars are made possible through generous donor support. If you'd like to contribute, please visit our website at autism.com. And now I'll turn this over to Dr. Lein.
Thank you, Denise. Good morning to everyone, or good afternoon, depending on where you're calling in from. I'm out here on the West Coast, so it's still morning for us. So today I'd like to bring you up to date on some of the current research going on, not only in my laboratory, but the laboratories of my colleagues across the globe. They're really focused on the question of how do environmental chemicals interact with genetic mechanisms to influence autism spectrum disorder, or ASD, risk. So one of the questions I'd like to tackle first, many of you are probably already aware of it, but it's still kind of a question out in the field, is why should we even consider gene-environment interactions in ASD pathogenesis? I think one of the most compelling reasons or bit of evidence that people use to really start focusing on the interaction between gene and environment is this incredible rapid rise in the number of cases of autism that have occurred since 1975 to the current day. This slide is up to date with the published literature, although I think the evidence is out there saying that the incidence of autism may even have been increased beyond what's shown here. But you can see that in 1975, 1 in 5,000 children were diagnosed with autism. And then in 2014, the CDC, or Centers for Disease Control here in the United States, released some startling data that in the U.S., 1 in 68 children are likely to be diagnosed with autism. And if we look strictly at boys, because there's a prevalence of this disease in boys, 1 in 42 boys are diagnosed with autism. This is just a remarkable increase over a relatively short period of time, which kind of argues against it being a solely genetic-based disorder. So what is the evidence that environmental risk factors contribute to ASD risk? Despite this increase, there's a lot of people who have really questioned whether environmental factors may be important. And that is really due to a number of factors. So the question has come up, how much of the increased prevalence of ASD represents an actual growth in the number of cases of ASD? Because over the same time period in which we've seen the increase in the number of cases, there's also been a dramatic increase in the awareness of autism as a disorder. There's also been improved detection and broadening of diagnostic criteria for ASD that likely contribute to this increased prevalence. In terms of this broadening of diagnostic criteria, this is also known as diagnostic substitution. It really refers to the phenomena that people are being labeled today as autistic who previously would not have been diagnosed as being autistic. They would have been diagnosed as something else. So these factors undoubtedly contribute to the increase, but are they the only explanation for the rapid increase in the number of cases that we see on an annual basis? A number of laboratories have scientifically investigated this question. I'm presenting here some data from that of my colleague at UC Davis, Irva Hirsch-Picciotto, and her colleague Delwich. It was published in 2009. And basically what they did was look at the annual incidence rates of autism based on the administrative database of the California Department of Developmental Services over the period of 1990 to 2006. What you see represented in this graph is the number of new cases per 10,000 child years, broken down into two age categories, children between the ages of 0 to 4 at the time of diagnosis and children between the ages of 5 and 9. Based on these type of data, they determined that there was a 600% increase in cases over this time period, or this time frame of 1990 to 2006. 24% of this they could attribute to an earlier diagnosis of autism. So autism was being diagnosed at a much earlier age than it had been previously. 6% of the increase was due to the inclusion of milder cases that would not have been diagnosed as autism at earlier time points. And 120% they could attribute to changes in diagnostic criteria. So at the end, this left 400% of the increased cases that could not be attributed to diagnostic substitution. This study has been replicated by a number of other labs using different cohorts of individuals across different areas of our country and abroad now. And while the numbers vary slightly as to what percentage of the increase is due to diagnostic drift as opposed to the percent due to other factors, does vary from study to study, but they are all consistent in finding that you cannot account for all of the increase in the number of cases strictly on the basis of changes in diagnostic criteria or increased awareness. So it appears that there are other factors at play here that we need to consider when we think about why it is that the number of cases have increased so dramatically over the last 20 years. Another reason that we think environmental factors are probably involved in the pathogenesis of autism is the fact that autism spectrum disorders really is a spectrum of disorders. So there's great clinical heterogeneity. So even individuals that have the same genetic background can have very different clinical manifestations. What you see on this slide are sort of the core deficits that really define ASD, and these include language disability, social communication deficits, and repetitive behaviors. So again, those are the core clinical features. But as many of you are aware, if you really look at the clinical picture, you see that there are intellectual disabilities that variably appear in the autistic population. There are also a number of irritability factors or other types of affective disorder sensory issues that appear in a subset of these children diagnosed with autism. There are a number of other comorbidities, sleep disorders, mood disorders, anxiety, hyperactivity, and attention that can appear in a subset of children. And then we have these other comorbidities that are not related to emotional, affect, or intellectual ability, but seem to be very distinct, and that includes seizures, gastrointestinal disorders, and immune dysfunction. So this is sort of the broad spectrum of issues that have been identified clinically in children diagnosed with autism, but not every autistic child will exhibit all of these phenomena. So again, great clinical heterogeneity suggesting that it's not just genes that are driving this, or that perhaps there are multiple genes that are driving this. So that leads us to the genetic evidence. What do we know about genes of autism? And there is evidence suggesting that genes play a role in ASD etiology. And so that is that if you look at monozygotic twins versus dizygotic twins, there is a much higher concordance rate in those monozygotic twins, ranging from 60 to 90%, depending on which study you look at, to a rate of 0 to 30% in the dizygotic twins. So this suggests that genetics are indeed important in the pathogenesis of autism. Another factor that points to a role for genes is the fact that there is a recurrence risk in families ranging from 12.9% to 18.7%. If it were not genetically linked, we would not expect to have this recurrence risk occurring in families. Then last but not least, more than 100 ASD susceptibility genes have been identified. However, these data not only argue for genetics playing a role in autism risk, but they also argue that there's got to be more than genetics. And that's because if you start looking at these ASD susceptibility genes, specific genetic mutations account for less than 8% of ASD cases. Not only that, but if you look at any given gene that's been associated with autism, that there's really an incomplete penetrance of ASD-linked mutations. So again, arguing that something else is interacting with these genetic substrates to really modulate or determine the final individual risk for autism. And so this is actually kind of an exciting hope that comes up in the field, because if we can identify what these environmental risk factors are, we might actually have a way to modulate risk. And that's because the identification of environmental risk factors for autism, in contrast to these genetic risk factors, which are currently irreversible, allow us to potentially modify risk. So what do we know about environmental factors? Well, we have a really rich body of literature that has informed us that there are diverse environmental factors that can interfere with neurodevelopment. So this is not strictly in the context of autism, but just looking at human brain development, we know that a number of environmental factors are important, and given the context, they can actually interfere with normal neurodevelopment. So we can break these down into three broad categories. There are psychosocial factors, there's maternal infection and inflammation, and then there are chemical agents. And so with respect to the psychosocial, maternal stress, nutritional status during pregnancy, child care, the age of the parents, and timing between pregnancies can interfere with neurodevelopment or alter or modulate neurodevelopment. And most of these have also been identified as potential risk factors for autism. There's a lot of interest right now in maternal infection and inflammation being environmental factors that alter or determine individual risk for autism. So these include factors such as radiation that can cause inflammatory events in the fetus and in the maternal environment. There's also physical trauma during childbirth, such as oxygen deprivation, which can give rise to inflammation. And then, of course, there's maternal infection, maternal illness, such as rubella, which can be mild, moderate, or severe. And then we come to this third category, chemical agents, which is really where most of the rest of my talk is going to focus. So this, again, is a very broad category. It can include prescribed drugs, such as anticonvulsants, antidepressants, anxiolytics. It can also include substances of abuse, alcohol, cocaine, methamphetamine. These are well known to interfere with neurodevelopment. Prenatal vitamins and nutrition can have either an adverse effect or a beneficial effect depending upon the levels that are present in the diet. And then last we have this category known as environmental chemicals. So these typically refer to substances that are inadvertently introduced into the environment, either through man-made sources or through natural sources or natural mechanisms. And another term that you often hear referred to or used to refer to these chemicals are xenobiotics. So what do we know about these environmental chemicals? Do they increase risk for ASD? This has been widely postulated, and we have many leads to chemicals that might be important in increasing risk for autism. But at the current time, I'd have to say that there are really few definitive hits. So what are some of these candidates that have been put forth? They include some of the classic environmental chemicals that we know interfere with neurodevelopment. So one category are heavy metals. There's interest in lead. You've probably all heard the story about methylmercury. I want to point out right now that trimerisol, which was the preservative used in vaccines, was not methylmercury, it was ethylmercury. Be happy to take questions on that later, but the methylmercury I'm talking about is not the type that was found in the vaccine. Also another category that has been postulated widely to be a risk factor for autism are pesticides. And there's three types of pesticides, chemical classes that have been looked at. This includes the organophosphorus pesticides. Examples of this are chlorpyrifos and diazinon. Also includes the organochlorine pesticides. Many of you might be familiar with DDT, which was the source of Rachel Carson's Silent Spring, but also dieldrine and lindane. Many of these are not in wide use anymore, although DDT is making a comeback because of mosquito-borne diseases. But they are very persistent in the environment. So we still have people in our country that have body burdens of these particular organochlorine pesticides. Pyrethroids, which are more of a modern pesticide, not as persistent in the environment, have also been linked to increased risk of autism. Now, again, we don't have definitive data that these are causing autism. In fact, by themselves, they probably are not causing autism. But there's enough interest out there, enough evidence to suggest that perhaps in combination with genetic susceptibility, these compounds might be increasing risk for autism. Another very broad category of environmental chemicals that have really risen to sort of the top of many people's lists of concern are the persistent organic pollutants. So as their name implies, these are compounds that are persistent in the environment. All of us carry body burdens of these, some of us higher than others, depending on what our exposure sources might be. Subcategories of these include the polychlorinated biphenyls, or PCBs. These have classically been thought of as legacy pollutants. They were manufactured until 1979, when their production was banned in the United States and worldwide. However, they persist in our environment, and as I'll get to later, we now have some contemporary, unintentional sources of current or contemporary PCBs. Another category are the polybrominated diphenyl ethers, or PBDEs. These are the brominated flame retardants that I'm sure many of you have heard of. And then last but not least, polycystic like aromatic hydrocarbons, or PAHs, which are produced when organic materials are burned or when there's combustion. And then more recently, there's been a lot of attention paid to a very complex environmental chemical mixture, which is air pollution, in particular either diesel exhaust or traffic-related air pollution. So again, we have a number of leads, but why has it been so hard to actually link any specific environmental chemical to increased risk for autism? And that gets down to the fact that, as I said a minute ago, that none of these environmental factors are likely causing autism in isolation, that they need to interact with genetic susceptibility factors in order to really have an impact on the risk for, I have here an NDD, which is neurodevelopmental disorder, but you could substitute that with ASD to influence the risk or severity of ASD. And another aspect of ASD that's really becoming more important, but it's been, I think, understudied to this point, is how these gene-environment interactions can actually influence treatment outcomes. So why is it that some kids with autism respond to one type of therapy and others don't? And so it might be that these gene-environment interactions also will influence that aspect of the disorder. And then superimposed on top of the gene-environment interaction is the timing of exposure. There's a long, rich literature in the neurotoxicology field that tells us that any given toxin may or may not have an effect on the developing brain depending upon the time of exposure. So there are going to be particular processes undergoing in the developing brain that will be susceptible to any given neurotoxin. And so whether the neurotoxic chemical has an effect depends on whether that developmental process is in play at the time of exposure. So another real big challenge to really definitively linking environmental chemicals to autism risk is the fact that not only do we have this gene-environment interaction to deal with, but as I indicated earlier, it's a messy genetic landscape. Hundreds of genes have been associated with ASD. And so each one of those genetic susceptibilities will probably give rise to a very different risk profile. So a person with one particular genetic mutation will have a very different risk profile for any given set of environmental chemicals relative to a person who has a different genetic risk susceptibility. So this is illustrated in this figure here, which actually comes out of an overview chapter that I wrote in a very good book that I recommend for anybody who's really wanting to follow up on this topic, which is Environmental Factors in Neurodevelopmental and Neurodegenerative Disorders, which was published in 2015. So what you see here are two graphs. If we focus first on the graph on the left, this is a classic sort of bell-shaped curve showing the distribution, in this case, of a neurodevelopmental disorder as a function of the percent of the population. And you can see that on the x-axis, we have the log or the concentration of the environmental chemical. So as the concentration of that environmental chemical changes, increasing from left to right, we have an increasing percentage of the population responding until we reach an apex, and then beyond that, we have a decrease in the percentage of the population that responds. So this reflects the fact that all of us have a unique genetic profile. So some of us will be particularly susceptible to this environmental chemical, which would be the people on the left of the TD50, which is the toxic dose at which 50% of the population will manifest the neurodevelopmental disorder. So people on the left are very susceptible. They react to very low concentrations of the chemical, whereas people on the right are highly susceptible, and they will, they're highly resistant and less susceptible. So it takes more of the chemical to cause an impact on that manifestation of the biology. So that's just looking at the population as a whole. We see a bell-shaped curve. Now, if we move over to the graph on the right, you can see our neurotypical curve is still there, but now superimposed on that are multiple purple curves, which refer to individuals with neurodevelopmental disorders. And because they have a diverse genetic background, we have sort of subpopulations within that population that has a neurodevelopmental disorder. And so some populations, because of their genetic background, are very susceptible to this chemical, whereas some populations are highly resistant. And so superimposed on the fact that we have hundreds of different genes that have been linked to autism, there are a lot of different chemicals out there, but now we have these different susceptibility profiles. So you can imagine that if you're trying to conduct an epidemiological study, which is typically limited in the number of individuals that are part of the study, a small percentage are going to express autism, and amongst that group that will express autism, we have all these different genetic substrates. So this makes trying to find a really hard and very robust association with chemical exposure extremely difficult. So these are some of the challenges that the field has faced in trying to really link a specific environmental chemical to increased risk for autism. So this is where people who are not epidemiologists, such as myself, who really work in cell and molecular mechanisms, have tried to provide some insight. And so the first question that people like me ask is, well, how would an environmental chemical interact with a genetic susceptibility factor to actually increase risk of autism or another neurodevelopmental disorder? I have to say at this point that this is still largely unknown. There are a lot of theories out there, and people are beginning to gather evidence to support these different postulated mechanisms for how an environmental chemical would interact with the genetic susceptibility factor. Some of the most direct mechanisms that have been proposed, of which there is some limited evidence, are that chemicals can actually cause mutations in the DNA directly. We know there are a lot of mutagenic chemicals out there. That's been known for a long time from the cancer field. And so there's a lot of interest right now in determining whether we can find examples in the human population or even in preclinical models of chemicals that can induce mutations that, in genes that have been linked to increased susceptibility for autism. That's a rapidly evolving field, and at the moment I actually don't have any specific examples to show you of that other than to say that people are actively exploring that possibility. The other field that's really exploded in the last 10 years is looking at the possibility that chemicals can induce epigenetic changes. I'm not going to spend a lot of time talking about this because that's a whole hour lecture in and of itself, but essentially epigenetic changes are changes not to the sequence of the nucleotides that make up the DNA. So the DNA itself does not change, but it's changes in how that DNA is expressed. So it can be changes in the methylation patterns of the nucleotides in the DNA. It could be changes in the histones that modulate the folding or the wrapping of the DNA. It can also be changes in what we know now are small non-coding RNAs that modulate how genes are expressed. So these are changes that will actually persist from one generation to the next generation, and that's the definition of an epigenetic change. And so lots of evidence starting to come out now that chemicals that we know can interfere with neurodevelopment can have effects on epigenetics, and that we do see changes in the epigenome between autistic patients and neurotypical individuals. So suggesting that this could be a very fruitful mechanism to explore to understand how it is that environmental chemicals are changing autism risk. So then the other mechanism that we consider sort of a direct mechanism of interaction between environmental chemicals and the genome would be a category known as heritable mutations in xenobiotic metabolism. As I defined earlier, xenobiotic basically refers to environmental chemicals, and it is well established that humans express genes that can metabolize these xenobiotics, in some cases making them more toxic, so they activate the environmental chemical to a toxic form, but in most cases to detoxify that chemical, so it's more readily eliminated by the body. There are examples of children who have mutations in the genes that encode for the enzymes that are very important in xenobiotic metabolism. This has been shown in a number of different cohorts now. What is not quite clear yet is whether those mutations increase their risk for neurodevelopmental defects caused by exposure to environmental chemicals. But again, a very active area of research, and at least biologically seems like a plausible mechanism to explain how environmental chemicals can interact with genetic mechanisms to increase risk for autism. All right, so this is a table that came out of this chapter that I wrote in the book that I referred to earlier that basically lists the number of mechanisms that have been proposed and the rationale for proposing them. Those in the white boxes we've already talked about. So again, these are sort of the most direct mechanisms by which chemicals interact directly with the genome to cause some kind of an effect that might increase risk for autism. But there are a number of new ideas that are out there, and these are rapidly evolving ideas as well, that are in the teal-colored boxes. So one of these is endocrine disruption. There's been a great deal of traction in this field lately. And the rationale is that we know that many neurodevelopmental disorders, including autism, are predominant in one sex. And this, and as we know, many hormones drive those sex differences in humans. These hormones are also required for normal neurodevelopment. So this includes not only the sex steroids, but also thyroid hormones, and that they have significant effects on neurodevelopment. That would be the glucocorticoids. So the theory is that endocrine disruptors, which are chemicals that interfere with endocrine signaling, may actually change neurodevelopment and may explain some of the bias that we see in one sex versus the other. Another mechanism that's been recently put forth would be chemical disruption of the gut microbiome. So there's emerging evidence that indicates the gut microbiome regulates host response to pathogenic microbial or xenobiotic exposures. So in other words, the bacteria in the gut can actually change how the host is going to respond, not only to the microbes, but also to environmental chemicals that come in, for example, through the diet. It's also known that the gut microbiota in children with autism differs from that of neurotypical children. So there's two ways to think about this. One is that the microbiome that's different in an autistic child will change the host response to that environmental chemical. Therefore, there will be a different effect of the chemical on the autistic child versus the neurotypical. The other thought is that the chemicals themselves may change the microbiome and therefore change neurodevelopment. So again, another area of active research. I think there's nothing concrete yet that's been published to really provide definitive proof of this as a mechanism, but it certainly is intriguing, and a lot of people, as I said, are actively exploring that. A third mechanism is immune dysregulation. As you may recall from the slide I showed previously, many children with autism do have immune dysfunction. It is also now widely appreciated that there is significant crosstalk between the nervous and the immune systems during development, and that this crosstalk is absolutely essential for normal neurodevelopment. It's also known that environmental chemicals can alter immune function. And as I said, there's increasing evidence of immune dysregulation in neurodevelopmental disorders, including autism. So potentially, environmental chemicals could alter the immune system during these critical periods when the immune system is regulating neurodevelopment. And this could set up the changes in the brain that are underlying autism. Again, not a lot of evidence to directly support that. There's a lot of ancillary evidence to suggest this is a biologically plausible hypothesis, and many people are pursuing it. And then the fourth mechanism that's listed here at the very bottom is the convergence of environmental and genetic factors on signaling pathways. So as many of you probably are aware, that there are a set of canonical signaling pathways that are critically important for the brain to develop normally. And what we now know is that many of the genetic risk factors for autism are converging on a subset of conserved signaling pathways that are critically important in neurodevelopment. We also now know that there are a number of environmental chemicals that influence exactly the same signaling pathways. So the thought is that these genetic risk factors converge on the same signaling pathways as the environmental chemicals, and it's particularly the pathways that are involved in synaptic connectivity in the brain, and I'll explain that in a minute, that may be the targets of these environmental chemicals. So the combination may push the organism over the balance to go into autism as opposed to staying on a typical neurodevelopmental trajectory. Okay, so I'm going to focus on that last category a little bit more. This is really where a lot of my research has focused, and I think a number of other folks around the world are kind of focusing on this as well. So again, we're looking at this idea that genes may interact with the environment to influence ASD risk, severity, and/or treatment outcome if both the heritable genetic vulnerabilities and the environmental chemicals converge on the same signaling pathway. So in other words, those genetic vulnerabilities may amplify the adverse effects triggered by the environmental exposure if they're both converging on these same signaling systems at critical times of neurodevelopment, and this can result in altered patterns of connectivity in the developing brain. So I've just thrown a lot of information at you there, so let's go back and dissect those different pieces out a little bit further. So first of all, why are we focusing on neuronal connectivity, and what exactly do we mean by that? And so there's a schematic across the top of this figure, panel A, that shows in a cartoon what happens in terms of neural development when that neuron is first specified in the developing brain. So we have a cell that is determined to become a neuron. And so as you can see on the far left of panel A, it starts out as a single cell with a nucleus. There are no extensions from that cell. As many of you are probably aware, neurons have a very unique structure which enables them to perform their function. And this structure involves the extension of processes from the cell body. And neurons can extend two types of processes, dendrites and axons. And early in development, they start out as very simple processes extending from that cell body. The dendrite forms the primary receptive surface of the neuron. So this is where signals will come into the neuron. Those signals will then be integrated in the neuronal cell body. A response will be determined, and that response will then be transmitted out to the next cell in the circuit via the axon. So the axon transmits the signal from this particular neuron to the next cell in the circuit, the downstream cell in the circuit, whereas the dendrite is receiving the information from the upstream cell in the circuit. Then as development continues, and we move to the right in this diagram, we can see that neurons undergo what is known as a dynamic state or critical period. This is a period of active change in these dendrites and these axons. They become longer, they start extending branches, and they begin to contact the cells upstream and downstream in the neural circuit. And there's a lot of change. This is a very plastic period. So processes are added, they're subtracted. Branches are added, they're subtracted. So this is very important to setting up the mature networks that are formed in the brain. And this is driven largely by activity. So there's going to be stimulus coming into the system, which we know as electrical activity or chemical activity. But basically this activity drives this process of these extensions coming and going. And that's very important, as I said, to basically shaping or pruning the final dendritic arbor, as we refer to it, or to the final axonal plexus. And then as you move further to the right, the system reaches a stable state where there isn't as much plasticity. The system is still plastic. It responds to, example, additional activity, and this is the basis for learning and memory. So this has to have some plasticity, but there's a low rate of turnover compared to what was going on during that dynamic state or critical state. And so if you look down in panel C, this sort of illustrates a little bit further what's happening in that critical period. So you can see an excitatory neuron there. This is a neuron that's going to release excitatory neurotransmitters, as well as what is known as inhibitory interneuron, which basically releases inhibitory neurotransmitters to dampen down the response in the excitatory neuron. So this balance between the excitatory and inhibitory input in the brain is very important to establishing these neural circuits. If we look at that excitatory neuron, we can see the green processes extending towards the top. That's the dendritic arbor. And they are forming contacts with incoming processes from the upstream cells. So we have a dark blue neuron extending its axons to form synapses or points of connection with the green dendrite on the recipient cell. And where you see those balls, that's points of contact, and these are known as dendritic spines. And under strong activity, those synaptic contacts will be strengthened. In contrast, if we look to the right, which is another incoming neuron in light blue that has weak activity, in this case, those synapses are not going to be strengthened, and over time will actually be pruned. And you can see that as you move to the right in panel C, we can see the stabilized synapses that were stabilized by strong activity versus the pruned synapses. Because there was weak activity, they were pruned away and eliminated. And so this is how this process occurs. Now, if you look back at panel B, we can see synapse number as a function of time of development. We have very few synapses in the human brain at early in development. This number starts to ramp up before birth, continues really taking off during childhood, and then during adolescence, there's usually a period of synapse elimination, and then when we get into adulthood, we're into what's known as synapse maintenance. Disruptions in any of those periods marked by the number 1, 2, 3, or 4 have been associated with specific neurodevelopmental disorders or neurodegenerative disease. With respect, or in the context of this talk, period 1 is really the one that's most critically relevant to autism. So there's now lots of evidence to suggest that a synapse formation doesn't go according to plan, or that somehow that trajectory is altered that might be the neuroanatomical substrate underlying many cases of autism. So that was a diagram. Do we have actual evidence from the clinics or from preclinical models, but mostly from the clinics, that this is true? And actually, yes, we do. So histological, electrophysiological, and functional imaging studies all point to altered neuronal connectivity in autism spectrum disorders. This can include things such as abnormalities in dendritic shape, which happens to be actually one of the most consistent pathologic correlates of behavioral deficits in neurodevelopmental disorders, including autism, but also functional imaging, which has illustrated both hyper and hypoconnectivity. And there's a cartoon illustrating that at the bottom of this slide. And this is actually taken from the paper. The citation is shown in the right-hand corner, but these are functional MRI studies looking at the connections in neurotypical children, which are shown on the left, versus children with autism. You can see there's a lot more connections that can be picked up by functional imaging in children with autism. So typically in autism, the feeling is that there's hyperconnectivity in local circuits within a brain region, and perhaps hypoconnectivity or not as much connectivity between brain regions, and that this might explain some of the clinical manifestations that we see in autism. So in addition to the clinical data, the genetic data itself also really points very strongly towards synapse formation and stabilization being one of the key pathogenic events in autism. So we see that many of the genes of susceptibility, when you start to categorize them by function, converge on signaling pathways that control synapse formation. So in this particular picture, which is taken out of a wonderful review by Ebert and Greenberg, you can see a presynaptic cell, which is the incoming cell on the top, and then the postsynaptic cell illustrated on the bottom. And the space in between these two is the synapse. So that's the gap between the pre- and postsynaptic neuron. Typically, in most situations, if you're looking at activity driving the formation of connections, that presynaptic cell will release glutamate, a neurotransmitter, excitatory neurotransmitter, that will diffuse across that synaptic cleft and bind to glutamate receptors on the postsynaptic side. There are three different types of glutamate receptors illustrated in this particular picture: the NMDA receptor, the AMPA receptor, and the metabotropic glutamate receptors. Not important to know the details for this, other than to know that there are different receptor subtypes that bind to glutamate. When glutamate binds to those receptors, it will cause an increase of calcium to flow into that neuron, also release calcium stores from inside the neuron, and this will activate calcium-dependent signaling pathways, which you can see in the red box there. This turns on a number of signaling molecules, which will then change gene transcription to then alter the structure of the neuron. There are a number of other factors here that regulate this process of synapse formation and synapse stabilization. Some of these that are represented in this slide have been linked to autism. So for example, neurexin and neuroligin, which are adhesion molecules that stabilize the interaction between the pre- and the postsynaptic cell, have both been implicated as genes of susceptibility for autism. We see also in the postsynaptic cell, Shank and Homer, they've been implicated as genes of susceptibility in autism, and then a number of other factors here as well. But I draw your attention to the calcium-dependent signaling, and then up on the far right, relin, which is actually an extracellular matrix protein, but also very important in synapse formation and stabilization. And it has also been implicated as a gene of susceptibility in autism. We're going to come back and talk about those in the context of environmental chemicals. But the key point here for you to take away is that the genetic studies tell us that synapse formation and synapse stabilization is a very critical endpoint in autism, because many of the genes that are implicated in autism really somehow are involved in synapse formation and stabilization. So this has led to the following sort of hypothesis that I think many investigators in the field do embrace. And this is out of a wonderful review by Levitt and Campbell, published in 2009. And you can see a neurotypical neuron represented in the far left. This is sort of a light blue color. So in this case, none of these mutations or copy number variants that are associated with autism are expressed in this neuron. So this neuron will go on to develop and not give rise to autism. And if I draw your attention down to the box at the lower right-hand side of the slide, you see sort of a two-headed arrow. So if you move towards the left, this is going more towards a neurotypical trajectory, and the individual will not be diagnosed with autism. However, if you move to the right, where the color changes from blue to black, this predisposes the individual to having a higher risk of being diagnosed with ASD. And so the blue versus the black indicates the either load of genetic susceptibility factors or hits by environmental factors. So in the blue end, no ASD end, there's a low load or no load of ASD genetic risk factors and no exposures to environmental factors that might predispose to changing these signaling pathways that are involved in synaptic formation. However, as you move to the right, either you have one or more genes being expressed that can predispose to autism, and then maybe superimposed on top of that are the environmental factors. So it's sort of a balancing game. How many risk factors are superimposed on the substrate of this developing brain, including both genetic and environmental? And at the top, you can see here an example where you have a genetic risk factor which changes the neuron from light blue to darker blue, and then that individual gets hit with an environmental factor that moves it farther to the right, increasing the risk for autism. So that's the model that many people are currently exploring. So as that as a background, I'm going to walk through two case studies pretty quickly of environmental chemicals that maybe illustrate this a little better, and also perhaps define for you why it is that many people are considering organophosphate pesticides and polychlorinated biphenyls as possible environmental risk factors for autism. So the first case study is really looking at the interaction between organophosphate pesticides, or OPs, as many people refer to them, and genetic risk factors. So first of all, OPs are a category of pesticides, as I talked about before. An example that many of you may have heard of is chlorpyrifos. This is a phosphorothioate type of pesticide, so basically a class of OPs that's been widely used for crop protection and used in flea dips and roach control sprays. The structure of chlorpyrifos is shown here, but the important part to know is that chlorpyrifos, like many of these OPs, is metabolized by xenobiotic metabolic enzymes in our body. And so chlorpyrifos itself can be oxidized by the cytochrome P450 enzymes to give rise to chlorpyrifos oxone. Chlorpyrifos oxone is classically considered to be the active or toxic metabolite. So chlorpyrifos by itself traditionally is not thought to be neurotoxic, although there's some evidence to suggest that might not be totally true, but chlorpyrifos oxone is indeed a neurotoxic compound. Chlorpyrifos oxone is detoxified by PON1, which is another one of these xenobiotic metabolizing enzymes. PON1 is the abbreviation for paraoxonase, and you can see that this detoxifies it to a compound known as TCP. So TCP is often used as a biomarker of exposure to chlorpyrifos, and this will actually appear in the urine of people who have been exposed to chlorpyrifos. So what's very interesting about this is that there are several polymorphisms in the PON1 gene in the human population, and these polymorphisms or variants of the PON1 gene have been shown to affect the catalytic efficiency, so how well PON1 can detoxify chlorpyrifos oxone, or to affect the expression level of PON1. So hang on to that thought. It's going to come back in another slide or two. So why are people focused on OPs? What's the reason for that? And I think large part of the reason is that these OPs are well known to interfere with neurodevelopment, and this is based on epidemiological evidence as well as mechanistic evidence in preclinical models. So just a quick summary of the epidemiological evidence. A number of human studies now, I just have a few reported here, but there's been quite a few now, have reported behavioral and cognitive problems in school-age children following chronic exposure to low-level OPs. Some of the first evidence for that is actually shown in this diagram here, and this was a study that was conducted in Mexico, where they looked at children who grew up in the foothills, which would be further removed from the agricultural fields where the pesticides were being applied, versus children who grew up in the valley, which is right in contact with those agricultural fields with heavy pesticide usage. And these children were asked to draw pictures of their family. And so you can see the pictures drawn by the 4-year-olds living in the foothills and the 5-year-olds living in the foothills versus those of their age-matched peers that were living in the valley. And clearly there's a difference here. The children who were growing up in the foothills and had presumably less pesticide exposure had much more mature and sophisticated drawings than the individuals or children who lived in the valley. So this is sort of the first evidence that potentially OP exposure may be altering neurodevelopment. So there have been a number of epidemiological studies that have followed up on that. A study by Brenda Eskenazi here in the Central Valley of California has shown that perinatal OP exposures may be linked to increased risk for autism. And then a number of other studies have shown that susceptibility to autism is influenced by polymorphisms in PON1, which is a key enzyme involved in OP detoxification, as we just discussed, suggesting a potential gene-environment interaction. So layered on top of this, there was this fascinating series of studies that came out looking at relin. Relin is a gene that's also been linked to autism spectrum disorders, and it's been shown that there are reduced levels of relin, messenger RNA, and protein in both the brain and serum of autistic patients. And if you remember a couple slides back, I had indicated that relin was important in synapse formation and stabilization. So the theory is that if you have less relin available, that will modify the ability of the system to form and to stabilize these synapses. Now, one of the things that's very interesting about the mutations that have been linked to autism is basically a number of repeats in the 5' untranslated region of the relin gene. If this is more than 12 of these repeats, this can actually blunt or decrease relin gene expression by 25 to 50%. So it was these long relin variants, the variants that had more than 12 GGC repeats in that 5' untranslated region, that were associated with autism spectrum disorders in an initial study, and then in 3 independent samples after that, but not in 4 of additional studies that came after this initial one. So this raises a lot of questions in the field. Is relin really a mutation linked to autism? So one interpretation of these, I guess it's 8 different studies, 4 of which found an association and 4 of which did not, is that there's non-replication due to genetic heterogeneity. An alternative hypothesis is the manifestation of this genetic mutation is strongly influenced by exposure to OPs. And this hypothesis, alternative hypothesis, came about because of the geographic location of these cohorts that were examined for association between the relin variant and autism. It turns out that the 4 cohorts in which there was an association found were living in areas where there was high OP usage. In contrast, the 4 cohorts where there was no association identified between relin and ASD were in areas where OP usage was significantly less. So following up on that study, a preclinical study done in a rat model showed that developmental OP exposure, specifically to chlorpyrifos, which in this graph is abbreviated CPS, can actually decrease relin expression. Remember that long variant that was associated with autism also decreased expression of relin. So the combination of this study that shows that increasing amounts of chlorpyrifos results in accentuated decrease in relin messenger RNA expression, combine that with that long variant, would suggest that the combination together has an amplification effect on the decrease in the amount of relin that's present. So you have a greater decrease in relin if you have both a genetic factor and an OP hit. So there was a group in, actually a number of people involved in this study, Persico and Bergeron, who said, let's try and put this all together into a model. And they realized that not only were relin mutations potentially important in determining response to OPs, but also polymorphisms in PON1. Remember that detoxifying enzyme for the OPs. And so the model they put together is illustrated here. And to walk you through this, basically we have a graph, excuse me, showing relin protease activity. Remember, relin protease activity is important for that synapse formation and stabilization. So as that decreases, you have a greater chance of having something go wrong with the process of synaptic connectivity. And then we have sort of a red bar across here, a red dashed line, and that is the threshold for, in this case, correct neuronal migration, but it could also be synapse formation. So if you fall below that dashed red line, you're at increased risk for having something go wrong in neurodevelopment. And then we have 2 colored squiggles here. So we have the pink squiggle and the green squiggle. The green squiggle would refer to the individuals that have the normal relin gene. In this case, they have 7 to 10 of those GGC repeats. in the 5' untranslated region. So this is the normal gene expression. The pink squiggles refer to individuals that have that long variant. So they have greater than 12 GGC repeats. And so therefore, they're going to have decreased relin expression, which translates to decreased relin protease activity. So we have two sides of the graph, the left and the right. If we looked at the left, these are individuals who have a polymorphism that predisposes them to high PON1 activity. Versus, I might have misspoke there. So the left has the high PON1 activity, the right has the low PON1 activity. So putting all this together, if we look first on the left side of the graph, the individuals with high PON1 activity, they get exposed to OPs, which is what the lightning bolt is. The impact of that is such that it's going to decrease relin protease activity, either directly or by decreasing the expression of relin, as we saw in the previous slide. So in both the individuals with a normal relin variant as well as the individuals with the long variant, we see a decrease in relin protease activity in response to OP exposure. But in both cases, these individuals, even though the levels are decreased, are above that threshold for having neurodevelopmental defects. Now move over to the right, and here you have individuals that have low PON1 activity, so they're not going to be as effective at detoxifying the OPs. So as a result, the OPs have a bigger impact. So even though the green and the blue lines start out the same level as the individual with the high PON1 activity, the drop-off after exposure to OP is greater because, again, they are not as effective at detoxifying that OP. Now, in this case, the individuals with the normal relin variant are fine. They do not go below that threshold. But the individuals that have the long variant of the relin gene, that decrease now brings them below the threshold, so they now are at risk of having something go wrong with neurodevelopment. So this is a very interesting model. There are still people out there developing preclinical models to really prove this model. The story has not been quite as clear-cut as it might be envisioned from this particular graph, but there certainly is evidence to support that this model is biologically plausible. I also show it because even though it takes a long time to go through it, it illustrates very clearly, I think, why it is so challenging to actually tie a specific environmental chemical to an increased risk for autism, because we have these differences in exposure but also differences in genetic substrate that are going to determine how the individual responds to that chemical exposure. Okay, so I have another case study. I see that we're almost at 11 o'clock, so I'm going to just kind of touch upon the highlights of this study. And this is actually a work that's going on in my laboratory in collaboration with Isaac Pesa, who's also here at UC Davis. And it's really looking at calcium signaling defects as a point of convergence in gene bioenvironment interactions. In this case, we have developmental neurotoxicity, but also it could be applicable to autism. So our hypothesis is that CGG trinucleotide repeats in the FMR1 gene, which is the Fragile X premutation, influences susceptibility to non-dioxin-like persistent organic pollutants, or POPs, and or cytokines and chemokines identified in the plasma of women during pregnancy that are at high risk for having a child with ASD. So we're actually taking our data regarding exposure to non-dioxin-like persistent organic pollutants and the cytokines and chemokines from plasma samples that are being obtained from the MARBLE study group here at UC Davis. And these are women, pregnant women, who have already had one child born with autism, and therefore they're at increased risk for having a second child born with autism or another neurodevelopmental disorder. And so we're studying what kinds of chemicals are in their blood and what kind of cytokines, and then trying to use that information to drive our preclinical studies, to mimic those kinds of exposures in our preclinical models. So we actually predict that these gene-environment interactions will converge on signaling pathways that regulate the patterning of neuronal connections that are established in the developing brain. So I'm talking particularly here about non-dioxin-like versus dioxin-like PCBs. Dioxin-like, just like the name implies, are like dioxin in their structure. They form sort of a planar structure, so all the atoms lie in the same plane. As a result, they're able to activate the aryl hydrocarbon receptor, which is the receptor that mediates the effects of dioxin. In contrast, the non-dioxin-like PCBs, because of where the chlorine substitutions are placed, cannot lie in the same plane. The atoms do not lie in the same plane, and so they're non-coplanar. As a result, they don't bind to the AH receptor. Historically, it was thought that dioxin-like PCBs were a problem, and we did not need to worry about the non-dioxin-like PCBs. We now know that's not true, and the non-dioxin-like PCBs actually are fairly potent developmental neurotoxicants. And this has been shown in human epidemiological studies as well as in preclinical animal models. What's really important to know is that while many people think that these PCBs are legacy compounds, we don't need to worry about them anymore because their levels have been decreasing in the environment since their production ban in 1979. There's been a number of studies that have come out, and this is showing one of the EPA studies, showing that actually polychlorinated biphenyl levels in the air in school buildings are much higher than the EPA level for safety. And it's now known that many of these are off-gassing from caulks and from paints in our school buildings. We also now know that a number of these non-dioxin-like or congeners are actually being produced inadvertently during pigment production process, contemporary pigment production process, particularly the formation of pigments that form these really bright colors. It's actually a colleague of mine, Carrie Hornbogle at the University of Iowa, can tell you the color of your room based on the PCB congeners that are in the air. So why are we focusing on these non-dioxin-like POPs, in particular the PCBs? They are still a significant concern. The orthosubstituted or non-dioxin-like PCBs predominate over those dioxin-like PCBs in both environmental samples and human tissues. And as I said, there's a number of contemporary unintentional sources of these non-dioxin-like PCBs that have recently been identified. And then the latest NHANES studies confirmed widespread exposure to PCBs among women of childbearing age in the U.S. And then the last thing that's, I think, very interesting is that a recent study published by another colleague here at UC Davis, Janine LaSalle, showed that PCB and PBD levels in brain tissues of individuals who are neurotypical versus diagnosed with autism or neurodevelopmental disorder, that those individuals with genetic risk factors for NDDs had much higher non-dioxin-like PCB levels in their brain tissue compared to neurotypical controls. PBDE levels were not different, but the non-dioxin PCB levels were extremely different. What that means, we don't quite know yet, but it does seem to be also tied to changes in methylation in the brains of the individuals with the autism. So another thing that's very interesting that we now know is that these non-dioxin-like PCBs alter endpoints that are relevant to NDDs and to ASD, in particular dendritic arborization. And this is some data from a preclinical study in which rat pups were exposed to PCB95, which is a non-dioxin-like PCB, in the maternal diet throughout gestation and lactation. And then we examined the dendritic arbors and the neurons in the brains of these rats at weaning. So on the left is a schematic showing the structure of the, for those of you who know anything about dendrites, the basilar arbor of the dendritic pyramidal cells. And we can see that there appears to be more dendrites in the neurons of pups that were exposed to Aerochlor 1254, which is a mixture of PCBs, than to the vehicle control. And that's actually illustrated in the quantitative data to the right, which I'm not going to go through right now in the interest of time. We can mimic this in tissue culture. So here we take primary neurons out of the rat pup brain, grow them in tissue culture, and expose them to vehicle, which is the control, versus PCB95. And we can see again in the pictures in the top left that PCB95 appears to be increasing dendritic arborization. And that is borne out if we look at the bar graph to the right, which is looking at total dendritic length. You can see relative to vehicle control cultures, which are the white bar, exposure to PCB95, which are the dark bars, increases total dendritic length at picomolar and nanomolar levels. So the levels are pretty low, and these are relevant to the levels of PCB95 that have been documented in human brain. I won't go through this data in detail. If anybody has questions, please don't hesitate to email me later. But this basically shows that in these primary culture hippocampal neurons, the PCB95 is indeed increasing calcium. And we now know through a series of studies that these non-dioxin-like PCBs are basically mimicking activity, which remember I told you would turn on calcium-dependent signaling pathways to control dendritic arborization. PCBs basically hijack that same signaling pathway, which increases signaling through CAM kinase, through CREB, which is the transcription factor, which then turns on expression of Wnt2, which increases dendritic arborization. This is interesting because, one, this environmental factor is mimicking normal activity to sort of modulate dendritic arborization in the developing brain, but it's working through the exact same pathway, mutations in which have been linked to increased risk for autism. And so this has recently been borne out by 2 recent genetic studies looking at pathway network analysis that have linked calcium-dependent and Wnt signaling pathways to increase ASD risk, and the references for those 2 studies are here. So in summary, what this is indicating to us is that if we go back to that diagram I showed you at the beginning where genetic susceptibilities interact with environmental chemicals to influence the risk and or severity of adverse neurodevelopmental outcomes, in this case, the genetic susceptibility are these heritable defects in calcium signaling, which could be a number of different proteins, all of which have been linked to autism. And this is interacting with these non-dioxin-like PCBs. And when the 2 are present simultaneously, that increases the risk for autism. So getting back to the model that I initially started this case study with, which is we're looking at the interaction between FMR1 premutation as one of those heritable defects in calcium signaling. Why do we want to look at FMR1? This is a CGG trinucleotide repeat or expansion mutation in the FMR1 gene. Many of you probably know FMR1 is tied to Fragile X, but there is a condition in which there's not a complete lack of FMR1 gene expression, but rather a change in FMR1 gene expression as a result of these trinucleotide repeats, in which the individual does not develop Fragile X, but does have a number of neurodevelopmental disorders. And it turns out that this is one of the single most common monogenic associations with autism. So roughly 30% of individuals that have an FMR1 premutation will have a co-diagnosis of autism. So one of the things that we know is that we knock in mouse model that expresses this FMR1 mutation, that they do have impaired neurodevelopment that manifests long before any kind of neuropathology becomes evident. There's altered neuronal migration, there's altered dendritic arborization. Shown in this figure, which I'm not going to really spend much time on, there's also changes in calcium signaling and altered electrical activity. So the strategy that we're using is we're actually taking iPSC-derived neurons from individuals who are neurotypical versus individuals who express this FMR1 premutation, growing them in culture, and then exposing them to non-dioxin-like PCBs to determine if the expression of that FMR1 premutation makes them more susceptible to the adverse effects of these non-dioxin-like PCBs on neuronal connectivity, dendritic arborization, synaptic density, and neuronal activity. So just to conclude then, what do these studies mean for parents and clinicians? Well, I think one of the take-home messages is that it would be prudent to minimize and prevent exposure to chemical contaminants during pregnancy or early childhood, that this may actually improve clinical outcome. So some common sense practical approaches that can be employed: do not use OPs in the home or the yard, consume organically grown produce. This has been shown scientifically to significantly reduce body burden of these OPs. To work with your local agencies to minimize use of OPs in public places and or to increase notice to the public of OP spray schedules and locations. Regarding the PCBs, it would also be important to work with local agencies to determine levels of PCBs in public buildings. In fact, it was parents working with the local chapter of the US EPA and then also the New York State Department of Health that really uncovered the fact that PCBs were present in very high levels in the air in school buildings. So parents can have a huge impact on this. In addition, keeping dust levels as low as possible is helpful because OPs and PCBs both adhere to dust, and to wash stuffed toys routinely. It turns out dust likes to collect in stuffed toys. Don't use brightly colored paints in your home. As I told you, PCBs are a byproduct of these really bright paint pigments. And then to limit dietary consumption of red meats and fatty fish, which while the fatty fish can have healthful effects, this is where PCBs tend to collect. And with that, I'll end with an acknowledgement of my collaborators and my funding sources and the people in my lab who actually contributed to the data that I showed you. And I'd be happy to take any questions.
Okay, great. Okay, thank you so much for that presentation. For everybody who's still here, we are recording, so This will be online most likely by the end of the day today. We also have a quiz that will be available after the webinar. So if you're looking for a certificate of participation, you can use that link in your reminder email once the webinar is done. I know we have time for just a few questions. So Dr. Lein, I'll read these to you and then you can follow up with some answers. Some of them are pretty complicated, so I'll just give you a heads up. I'll start with a fairly easy one. This first person is asking about prenatal testing for environmental toxins in a mother who is pregnant, but I think they're also interested in environmental testing for their home. So do you have any recommendations along those lines or just sort of common tests you're aware of?
So actually these tests are not common, they're not cheap. They are out there, they are available, and I would actually direct anybody who's interested in this to a website that Martha Herbert has been maintaining. Martha Herbert is a pediatrician who's gotten very involved in environmental factors and autism. I can't remember exactly which institute she's affiliated with. I think it's Harvard. She's in Massachusetts, but she maintains a wonderful website. And actually what I can do is email Denise after this is over with that web address if anybody's interested. But she provides a lot of practical information on what types of testing you might consider and what it can and can't tell you.
Okay, great. The other part of that question was just about environmental testing for the home. So do you have tests like that that are probably not affordable based on what you said?
They're not routine, and typically you need to contact, I'm not aware of any state health agency that has any kind of routine testing available, but most states will have a university that has an environmental health center of some sort that will either enroll you in a study or provide some limited testing, or at least be able to suggest where you might be able to find somebody who could do the testing for you, and again, help you with interpretation of the data.
Okay, the next question, this person's asking, are any researchers looking at other health issues in the Relevance studies, regardless of whether the subject has expressed autism?
Yeah, so there is quite a bit of work going on clinical manifestations associated with Relin. So Relin really kind of became a hot topic because it's associated with some lissencephalies, which basically are clinical syndromes that result as from abnormal migration of neurons. And so that's really where Relin was discovered to be a gene that was really important in neurodevelopment. So there's a very active area of research focused on that. Whether there's anything beyond that, I actually don't know, because that's a little bit outside my area of expertise.
Okay, great. This person is just asking about things that people can do in terms of detoxification just through diet and through selection. I know you've talked about controlling the environment, so being sure to advocate at school, talking to administrators, and being sure to check on buildings. Is there anything that you're aware of in terms of nutrition? I mean, would this be something we'd point to Dr. Herbert's site again?
I would, yes, absolutely point to Dr. Herbert's site, because she does talk about nutrition quite a bit. And I do think that there's pretty compelling evidence that if you can afford to switch to organic, that does decrease your body burden of pesticides pretty considerably. Not everyone, of course, can, but just simply even washing your fruits and vegetables very thoroughly before eating them, and also peeling the outer skin off, can really significantly reduce pesticide intake. So I think those are two approaches that you can use to sort of modulate your body burdens. With respect to the persistent organic pollutants, that's a little trickier because they're lipophilic, so they tend to accumulate in fat depots in everybody, and it's just hard to dislodge them from that. So I think, again, switching to a healthy diet, to a clean diet, and I mean, everything I've read has said a well-balanced diet is your best, protection against a lot of these things.
Okay, so I think we have time for just one more question. This last question is about, you touched a little bit on the difference between ethyl mercury and methyl mercury. So that related to the mercury that used to be in the thimerosal, or is actually still in thimerosal. So if you'd like to maybe talk about that just a little bit more, about the differences and what it means.
Yeah, so I think one of the areas of real confusion is there's no doubt methylmercury is a developmental neurotoxicant. But the form of mercury that was in the vaccines, thimerosal, is actually ethyl mercury. And while it itself can also be neurotoxic, because of its differences in PK properties or pharmacokinetic properties, it basically doesn't distribute to the brain to the same level that methylmercury does. And so when people have actually done very detailed distribution or disposition studies in preclinical models, but also in humans, they have found that if you took the same amount of methylmercury and ethylmercury, very little of the ethylmercury actually gets to the brain. So I think that's one of the main reasons, of course, in all the epidemiological studies that have been done, that people have said that for the in general, thimerosal probably is not a huge risk factor for autism. It's because of the form of the mercury.